as Dorothea pointed out, like the focus of today is to interpret the soil monitoring. So just to get us focused here in terms of soil, I always like to start with a quote. This is from Wendell Berry, and it really gives us the whole global picture in terms of what soil means to our lives and in our human and non-human communities, and that caring for the soil is really an important aspect of our stewardship. So just to come back to that thought and to get as grounded in terms of the soil being the connector of all of those parts. So before we get really into a big headspace, I just want you to, consider you're sitting around the campfire and you're reflecting, or you're sitting here looking out the window, and just to maybe jot down something that stood out for you during the monitoring visit. And then, jot that down, and maybe at the end, we'll revisit that question, maybe in a bit of a different format. So it doesn't have to be complex, just, what stood out for you during the monitoring visit, and we'll refer back to that. And also, again, in asking you to make some notes, while we're going through this, we will be talking about some constraints and what that process might be referring to, and what adaptation you might be able to implement to address that constraint, and some notes. So maybe you have your own system. I'm just offering this as something you might start to take some notes on. If we don't take some notes while we're looking at this information, we kind of lose out on that opportunity to be able to implement it into our management plan. So our objectives for today in terms of soil health monitoring is get an appreciation for what the indicators mean. So we'll be looking at the overall description, and then what is the story that those indicators tell us? They, telling us about how we're interacting with the soil in past management, current management, and how can we, adapt that towards guiding our management towards regenerative outcomes. Regenerative outcomes being, improving that overall soil function, improving overall soil health. And then finally, being able to reflect on the process of collecting, monitoring, and looking at it, and being able to discuss it in a group. How does that catalyze the community of practice in regenerative organic farming systems? So us as a community, how can we, further value and utilize the monitoring as a way for the community to move forward? So just touching on the principles, I'm really going to touch on the first principle about context over and over again. With relationship to the soil, you can also think of it in relationship to where you're at now and where do you want to go and how you're going to get there. The other principles are, minimizing disturbance, keeping the soil covered, living roots as long as possible, adding diversity or maintaining diversity, and integrating livestock. And then the 3 rules of adaptive management. These are, universal rules in terms of compounding or cascading effects. We try to find ways of using this in a positive way. So, an example might be if we're, able to, compounding and cascading effects means if there's an action, there's a subsequent reaction, and oftentimes this has, further reactions. So we're trying to find ways that we can reduce negatives and add positives. So an example of that would be if we are making an adaptation to our rotation or a cover crop mix, that could have a cascading effect in terms of the soil physical qualities, in terms of reducing bulk density, and that in turn is also related to perhaps even at the end result of having better nutrient density. So that would be advantageous to us all, particularly in those that are grazing livestock. Rural diversity comes up again. It comes up 3 times in the principles, the rules of adaptive management, and in the 4 ecological processes, because it is so important. Ecological systems do not exist in monocultures. They exist in diversity because that redundancy, meaning there's several species or organisms that can do a certain job, and in certain situations they can shift a focus, and that diversity allows resiliency and the ability to come back from a harsh environmental or physical impact. So adding more species in our crop rotation is one of the first steps that is easier, or one way we can fill a gap. Underseeding or interseeding is another way we can add diversity. And then the other rule is disruption. So if we continue to do same thing in the same pattern, it creates an impact. And an example would be if you use the same rotation with all spring planted crops, you end up with a certain weed ecology. If you disrupt it by doing fall crops and spring crops, your weed management will change, because the weed seed bank cannot adapt to that sort of disruption as well as if it's always the same. And then the 4 ecological processes, in our assessment and our monitoring, we're trying to determine which of the 4 ecological processes are not functioning well, and they are interrelated. They're not, distinct and unlinked to each other. And so by focusing on, water infiltration to have an indication of how well the water cycle is working is a pretty direct measurement. And the biological assessments, giving us a more direct measurement of biodiversity. We have the soil test from A&L, which gives us the mineralogy or chemistry analysis, but we're also focusing on some other nutrient components, and then we're looking at carbon levels. So we're trying to use those monitoring levels so that we can focus where the management change needs to occur. And then we're trying to fit this all into the management approach. So that's stepping back in terms of getting us, situated where the presentations were last year, just trying to make sure that we touch on that again so that we can develop appreciation of where that monitoring is and how to use it. So I see that John is here, and he can, likely well remember this particular exercise. So everything we're talking about today, there's like a worst case scenario on the left where, the soil or the flour falls apart, and on the right side where this soil sponge or the bread can support the tractor and the greatest difference here is the bread represents a sponge with life, and the John Deere tractor is in bit of a mucky situation. So just trying to give you a visual difference here in terms of some of the aspects we're going to look at. And the monitoring that we're going to first touch on is the physical quality, which you can really appreciate. There's a big difference here. So in terms of the monitoring, most of you are aware that we were using managed areas at best and challenging our worst case scenario. And in some cases, those people who are in the rock certification were also adding a representative because that's a requirement. And we compared that to an unmanaged area. And there's, questions around why we would include an unmanaged area, and hopefully looking at the results, you'll get a further appreciation. The unmanaged area gives us an idea of the potential. In many cases, the unmanaged area has not been any management done to it for quite some time. So the distinct difference between the unmanaged area and the managed area is the management. And when we come down to all of these monitoring indicators, it's all very management dependent upon how you can get your better results, your worst results to your better results, or something closer to unmanaged area. So there are analysis here, samples that are sent to the lab for soil, for the physical qualities, the chemistry and the biology. And then, in many cases, there were plant samples where bricks measurements were done in the field and also sent to the lab tissue tests. So parts of the plant were sent to the lab to get an idea of the nutritional status. And what are the minerals that are present in the plant is a good indicator of the difference between what the soil potential is and what the other characteristics, quality of the soil, environmental conditions, and what the plant was able to receive. So we now have a combination of lab analysis and field observations, and what is distinctly different when we put this into the context of how to use the information is being present during the monitoring will give you the opportunity to instill a little better understanding of what that monitoring is about. But because you are in your management scenario, and you know what performance is going on with your management, you can link those outcomes, especially the numbers that you see from the lab, to what those outcomes are. And also by doing this with many of you in the same eco-region, you can develop an understanding of what would be the potential within your ecoregion, because you're getting this similar, not, there are some distinct differences for some of you, but you're getting similar environmental conditions, moisture, temperature, and what would be different is your management. And also, it gives you a language to discuss, which, other people may not be particularly focused on. And this, in our community of regenerative organic oats, gives you an opportunity to start using those monitoring outcomes in your language, provided we can all together make this an environment where we can be comfortable with curiosity and how those results, what they mean for your management, or how it might have meant for somebody else. So the first place I will start with is the soil texture. And this is one of the inherent characteristics. And the soil being made up of, three very distinct fractions of different size and different behaviors. The sand being the large, or what is more or less called the coarse material, and the intermediate being the silt, and then the clay component. And how many of these monitoring indicators that we look at respond, change, is really important, that you are comparing, in the first case, are you comparing your best and worst on unmanaged? Do they have similar percent sand or similar percent clay? And then you can say, well, now my comparison can be valid because at least from the starting point, they were similar texture. So this is a representation in terms of the soil texture classes. In your report, you'll get, so these are extractions that were done in the lab, and you'll get a report in terms of what is the percent sand, silt, or clay, and then based on that percentage, there'll be a nomenclature or name in terms of what texture class that is. So for the majority, just, for most of you, you're in the 40-60% sand range. And if some of you have ones that are different to that, then you can expect you could have different outcomes. So when we talk about organic matter levels or targets, it's very different for a sand, primarily a sand, than a clay texture. And I think in one of the charts that you were given, it gives you actually some ranges. And one of the other measurements is bulk density. That is dependent on texture, but in the end, it has a lot to do with our management in terms of, how the equipment is passing over the soil, the weight and repetition, the snow load in terms of the weight of the snow, how much tillage actually can, in the upper layers, will decrease the bulk density, but if the tillage forms a compressed area, that's what we call the plow layer. So plant growth is restricted. As you can see, the roots in the depths of the same core of soil are greatly reduced once the bulk density increases. So this is a weight of soil per certain volume. The bulk density is also used in the calculation of organic carbon. So if somebody wants to actually make carbon assessments, it has to be corrected with using bulk density. So one of the ways, and that was done in the lab, but one of the ways we do it in the field is with the penetrometer. And so we're finding where in the depth of soil, as we push the probe through, where do we find resistance? And when we get to 300 psi, it's an area that'll not only restrict roots, but it'll restrict the air exchange and the moisture. Sometimes there's also initial crusting on the soil, And when you're working with a tiny little seedling that doesn't have as much power as a mass of roots, then a lower PSI, at 200 PSI, is considered to restrict germination. So in the Care Labs protocol, they had asked us to measure the PSI at different depths, and this is to give them an idea of the species diversity or species richness at different depths of soil and at different compaction levels. This penetrometer is a point in time, and you want to look at the relative difference between your different management areas. The other field observation is if there is soil crusting. So this cracking or crusting, the fine particles, and we also see that in the water infiltration, whether they're swirling around and clouding up these fine particles on the surface, they're bare, they've had some tillage, and whether there's evidence of erosion or wet water has been ponding is another observation. So these are looking at symptoms of management, but we need to also get at the root cause of that, and where do we address it. So definitely looking at this would be, the soil is bare at this time, so it's very exposed, and so when there was a rainfall, there could be erosion, and after the rainfall is gone, the crusting happens, and the next rainfall, a lot of the moisture can't go in. So if you don't have moisture going into the soil, it takes the soil, it moves off and it takes the soil with it. So one of the principles to incorporate here would be to make sure the soil is covered and having roots, living roots all the time. So the other infield measurement, and this directly relates to the water cycle, is the water infiltration. Again, it makes, because we already pointed out there's a difference in how the pores are being held closer together when there's less, there hasn't been much rainfall. We look at the unmanaged to the managed area, and we use 3 rings, and that gives us an idea of the variability. And we also try to use 3 pores of an equivalent of 1 inch of water, and we time that to how long it takes. And then we also take the ring out afterwards to see what the soil looks like, and basically, does it look like chocolate cake, like with lots of pore space, or does it look like pudding? And this water infiltration has over and over again been one of the big aha moments in terms of how well unmanaged areas, how quickly they infiltrate, and how little variability they have. But also, while we're looking at this, this one in particular, the water is not clear, it's cloudy, it's picking up a lot of particles that are swirling around. And when those particles eventually settle, there's a high probability, depending, on other conditions, that they will eventually form a crust and to seal. So this is a really nice visual experience in terms of, well, if a rainfall happened at this point in time, can my rainfall be effective? Can I take in, that really desired 1/4 inch that I'm looking for? Am I going to effectively be able to infiltrate this? So water infiltration is a great one to show the differences. So one of the other ones that we sent soil samples off to the lab was for wet aggregate stability. So it's an indicator of the soil structure and its resistance to intense wetting events such as heavy rainfall or irrigation. It's presented in a percentage. And so the sample of soil is placed in a sieve, and those soil aggregates are dry, and they're of uniform size range, and then a column of water is placed over top, and the difference between what stays in the sieve and what washes away to the total soil is what gives you the percent. So this is you know, a way of actually determine if the spores are going to resist the rain or irrigation. Can the soil keep the pore space open, and can they keep the soil in place? So if you look at your unmanaged and your best and worst, see what the difference is in terms of wet aggregate stability. We're looking for a higher percentage, which means a higher percentage of the soil particles stayed together on the top sieve and didn't wash away. And this would relate to the needing to add more carbon and microbes. Those are the pieces that hold the pore space in place. So the carbon is what is the quality and quantity of carbon that you're putting into your soil or on your soil, and then the microbes are creating the glue parts. We call them, some people call them the snot, slimes, and glues to keep those pores together. And then also less disturbance, because tillage and other activities on the field will really change the capacity of that pore space to remain intact. So here's a photo of what it would look like after the aggregate stability test has been completed on the sieve, and then going back to the picture of the bread and the flour. The difference here is adding life to the soil, and that's the carbon material and the microbes that are taking the plant residues, the compost, the manures, the roots, and they're shredding it and taking it apart and digesting it, and then also synthesizing it and putting it back together. And I think the other one that we can visually look at in terms of how this is happening in the field is the dreadlocks, or looking at the roots in terms of how much of those micro aggregates are actually sticking to the roots, because we have a great carbon pump that's happening, with the photosynthate going into the root zone, and an invitation for those microbes to come in and digest those nutrients, and then make those aggregates form. And it's the process of those aggregates forming and the microbes synthesizing it that is going to lead to long-term carbon storage, and also short and mid-term food for the microbes, themselves, and also for the crop production. So it benefits you in your crop productivity and profitability, and it also benefits the soil health. I'm going to get to the other part of carbon as we go along, so I'll come back to the carbon cycle and those metrics. In the field, we've been measuring the bricks level, which is again a relative measure. I think I heard a story, maybe Larry, that or we can relate to the story in the field day of 23, where we used the reflectometer to measure what was the impact of using an amendment in the crop, and it was pretty astounding in terms of the sugar levels that were in the plant, comparing the treatment to the non-treatment. So that's another good way of measuring. So the sugars are the solids that the light is being that is refracting the light in the refractometer. So when you put the sap on the lens and shine the light through, the more sugars, i.e., the more minerals you have in the sugars, the higher the Brix reading will be. Yes, that's going to be depending on the time of day and how diluted those sugars are. So it is very much a relative measurement. And this year at the field day at the Shill farm, using the refractometer was another big aha moment, because Allison commented that, she didn't see any grasshoppers, and grasshoppers had been quite a bit of a problem with drought situations. And so we know that grasshoppers can't digest high sugar diets because they have a very simple digestion system. And the refractometer reading was in the zone of 12, which was really excellent. So it's another tool you can carry in your pocket and use frequently. This other one on the right is more or less looking at plant health, but we also use the tissues, taking samples to not only just diagnose plant stress, but also getting an affirmation of what the nutrients are within the plant. And it takes some finesse in terms of, understanding why the levels are where they are. One of the important factors to take into consideration is the plant partitions all the various minerals in different plant parts. So, you comparing a similar plant part in the period of time of, where it is in its vegetative state or where it is in its reproductive state, because as the plant goes through its life, it partitions nutrients in different parts of the plant. And a really obvious way of, it doesn't take much more than a shovel to go to your field every time you're at your field, is to just check what the feel of the soil is, what the color is, and what the smell is. And so on the left, you can see this is a little more compacted and blocky feeling. You can actually see some layers of where the compaction might be. And then this is something that would be a bit more desirable. You can actually see the residues still in the soil that are being digested. So I don't think we actually consistently did, a comparison and show that in the chart in terms of unmanaged or managed areas, but you can, make your database a little more robust by doing those comparisons. And then also, combining some of these tests, if you took a plant that has been successful to grow in this soil and measured the bricks, and a plant that's in this soil, or, you usually tell when you're in the field in terms of a plant that's doing well versus a plant that's not doing well. And then just dig a little deeper and see, are the soil conditions looking really different in terms of soil color, feel, and smell. And I should also not forget soil depth. So, I mean, prairie soils have wonderful depth, but there has been erosion over time. So, be cautious of where in the landscape you're measuring, uh, because, there is a tendency that topsoil, the top horizon, the darkest horizon, is likely deeper in the areas where soil has moved either by water erosion or tillage erosion to the lower areas. And it might be in your fence rows, and we've had that experience when looking at some fields. Um, and it might be a lot more shallow on some of the knolls. And then just looking at what kind of macro life we can see in the soil, pulling it apart, those that we don't need a microscope or maybe just an eyeglass to see in terms of worms or, various beetles, worm channels. Those are all observations and that you can use, and again, use them as a compare and contrast. To try to explain the results in one area or one field versus the other. And we've been really purposeful in terms of choosing that, why one field, using a best to a most challenging, really purposefully in terms of giving you a context in your farm, in your fields, in your management to see what the outcomes are. So I've been talking a lot, and I'm gonna move on to the soil chemistry. Is there a question so far? Everybody, it's okay, Dorothea, to move on, or do you want to?
No, it looks good, Ruth. I don't see any questions coming up.
Okay,
So let's head right into soil chemistry.
okay. So perhaps this is a little area in terms of what you may be more familiar with or you would like to be more familiar with. Um, and it's certainly an area that taking soil tests that has been utilized with fertilizer suppliers, and I've heard many organic farmers say, well, I'm not sure that I can use those with a lot of confidence because they don't really fit my situation. But it is, I assure you, it still is very important in terms of having a basic understanding of what these um, chemistry, how it works and what it means and how you can utilize it. So after we've looked at the, comparing the soil texture and looking at the cation exchange capacity, those things are fairly constant if we're measuring or we're taking soil samples from the same soil. Um, one of the ways that your cation exchange capacity could change is if there's been a lot of disturbance and you're into a different type of soil. But so um, that's not usually the case. So the next thing in terms of getting an overall idea of the context of the soil that you're working with is the pH. And pH is a logarithmic scale, means that the difference between a 1 and a 2 is a factor of 10, not 1. And it gives an indication of the balance of hydrogen ions or hydroxyl ions in your soil. Okay, so what does that mean? It means your soil is either alkaline or acidic, and there is an optimum range, and soils from the results here tend to be, you know, in many cases in the optimum range, which is in that, 6.4, 6.8, 7 being neutral. There are a couple of results that I've seen that have been down in this 5 range. And I would say that that is an area that you would seriously give consideration to monitoring, and even considering how you might be able to change that. And I do appreciate that in really broad acre management, it would be perhaps prohibitive to be able to put on a neutralizing agent, something with calcium carbonate that would move you from a 5 more towards your target. But this would be one of the limiting factors that changes many many of the nutrients and how they will be available to the plant. They also change the biology community, and the biology community can actually help to enhance movement of the pH scale. So look at that and compare your pH that you're seeing in your best fields to your most challenging fields and your unmanaged area. So as I said, the pH up this scale tells you the pH here of 7, which is neutral, and that's where there's not there's a balance between the alkaline and the acidity factors, but as you move away from neutral, it changes the availability of all of the macro and many of the micronutrients. Particularly phosphorus, which is more sensitive to the pH change, and where it can become unavailable quite quickly when you get to the acidic zone and in the alkaline zone. So this shows the macronutrients, so those nutrients that are needed the most. And when we're looking at the rest of this, we're just going to be focusing on potassium, calcium, magnesium, and the rest of the micronutrients. They are called micro because they're needed in small amounts. It doesn't, there are toxic ranges and there are insufficient ranges, but particularly phosphorus, nitrogen, those are the ones that are needed in larger quantities, and they are the energy source, and we want to address those first. So we would get into more focus on something like boron if the tissue result would indicate that could be addressed. But in the overall focus on soil health here, we look just at the macronutrients. So the next thing that really gives us a context is the cation exchange capacity. And I find prairie soils a lot more complex to understand in terms of cation exchange capacity, because in many cases I see the cation exchange capacity very, very high and not well related to the actual texture of the soil, where in our Ontario soils that are much more calcium based, and formed in the way that they have, not primarily grasslands. I see different ranges, and so the cation exchange capacity is basically how the cations are able to move on and off the clay particles in the soil. So I really like this presentation because here's a clay particle, and remember that's the smallest portion, um, smallest size particle, but it has a greater influence in terms of its electrical charge. And there's also a cation exchange capacity in the organic matter portion, which is a smaller portion of the overall volume of soil. But it also, because it has a cation exchange capacity and the other functions that it does, also has a influence on nutrient availability. So there's the clay particle with the negative charge and the cations with positive charge, which includes, and they are also of different molecular weight, but let's just focus on the electrical charges here. There's calcium, magnesium, both with a 2+ charge, and potassium with a +1+ charge, and then there's the hydrogen. And because of their charge, they will stick to the clay particle, and they bind to the particle, and when they're released, that's the cation exchange. So we're looking at the ability of the soil to exchange those cations. And roots don't consume the soil as a solid. These minerals, and they're not singular, they're complexed together, but we're looking at in this sense in terms of being singular. They need to go into soil solution out here before they can be absorbed by the roots. So the roots don't gobble up rock, they suck in water that has those cations. And the ability for that clay particle or the soil volume to let those cations be removed to allow the root to take those up is the cation exchange capacity. So the bigger the number, the higher the capacity for that soil to be able to give up those cations. However, when it has a high cation exchange capacity, it takes a bit longer to fill that up. So a soil that has a sand texture will likely have a lower cation exchange capacity. So it, other factors change the cation exchange capacity with, the type of clay it is and the silt, a portion. In exchange, the root hair is giving up the hydrogen. And this is also on the clay particle, and this also has an influence on the has an influence on the acidity and the cation exchange. So when you add fertility, so if you're adding calcium, it'll either go in solution or it'll bump off some of the other cation that's sitting on the clay particle and give it up to the soil solution that allow the plant to suck it up. So again, if you're comparing your soil particle, sand percentage, and your cation exchange, and then you can make comparisons in terms of what impact your management has on all the other factors. So this is just another way of showing, there's a clay particle and the cations have the charge of the positive on the clay is sticking to the charge on the cation, and they come on or off depending on the cation exchange capacity, and then the plant can take it up. So clay soils tend to have a higher cation exchange capacity. The cation exchange capacity in your soil report is not directly related to soil texture. It is a benchmark calculation based on when they take the soil into the lab and they do an extraction and they extract the calcium, potassium, magnesium, hydrogen, and sodium, and they add those up and they get the cation exchange capacity. So you're wanting to make sure that you compare your soils that have similar cation exchange capacity. It also has an influence on the targets that you're setting for your macronutrients. So if you have a lower cation exchange capacity, you have a lower target for your phosphorus, your potassium, your magnesium, and your calcium. If you have a higher cation exchange capacity, 26 and plus, then you're into a lower range in these cases. You have to be careful about the phosphorus because your target is an optimum range. Too little and you don't have enough productivity, and too much, you're going to be creating potentially an environmental impact. So that phosphorus gets out into the waterways and creates reduced water quality. Having too much phosphorus also inhibits some of your other nutrients, particularly zinc. I don't know if many of you, doing tissue tests have ever come across, that scenario. So in understanding the numbers and whether or not you are close to your target, these are the targets, the ranges potentially. And in this case, we're using the P-Bray, not the bicarb number, because generally you're in lower pH and a different type of parent material. So that's on a ppm basis. So the ppm gives you the number of that mineral that you have in the whole piece, and the percentages gives you how are they distributed. And on percentages, you get even a greater appreciation of if you have a higher level of calcium, it's going to impact the availability of some of your other nutrients. The potassium and magnesium ratio tends to be more related to how well the plant can get magnesium or can get potassium, and it also is related to soil health. I should point out that phosphorus is the predominant energy source for plants and most mammals, well, all mammals, even probably some of the microbiology, where potassium is related in respiration and that respiration has, because you're exchanging carbon dioxide and oxygen, has also an influence on soil health. That's getting further into the weeds than we need to now, but if you're looking at your test and trying to find value in them for, determining if your amendments are being adequate, and if there's an adequate mineral nutrient there for your crop productivity, these are the ranges that you should be looking for. And then, of course, see what the numbers are in terms of your best and worst, and trying to find out if any of these that looks like a limiting factor explains why you're best and worst. So predominantly we have, the agriculture sector has looked at chemistry as a way, so adding soluble fertilizers as a way of boosting productivity. And when we start to talk more about organic matter, we'll see that that hasn't necessarily bode well for the overall soil health. But as you start to look at the various results, you'll see that there are other factors that we're looking at in terms of the soil health. It could be compaction or water that does not explain the differences, and so therefore a deficiency in the minerals is not where we should be going. We should not be trying to address a challenge in the water cycle with nutrients. So we can't push productivity when the physical factors and the water cycle are not working. So I think the more exciting part about soil health is looking at the underappreciated part is with the carbon component, and that's where we're going to step into the soil organic matter. So soil organic matter on your data sheet there is expressed as OM and a percentage. So there's a big volume of soil, and a small volume of that is soil organic matter, and so the number is a percentage of the organic matter that is the whole volume of soil. And generally it's a test of, we dry it down and burn it and then measure how much carbon was lost. And it is a challenging indicator to use in terms of fully appreciating what management is actually contributing to the organic matter, and how we can change it. So we're going to talk about the legacy soil organic matter versus new organic matter. Which numbers are better indicators for slow movement versus more responsive movement. We're going to talk about quantity and quality, and then it all comes down to balances of addition. So additions of carbon in the form of root exudates, the carbohydrates that are pushing through your plant material into your root zone, the root mass that ends up in your soil and is broken down by microbes, the debris that's left over from your crop versus the crop you're taking away, and the residue that you might be taking away, or the residue that oxidizes. And that's the balance in terms of how can you increase the carbon levels in your soil, and the carbon levels in your soil being a prime food source, and then a catalyst for lots of other activities in terms of soil health. And it's one of the more challenging pieces to pull apart because I would say in terms of soils, this is the part in terms of knowledge that has moved more quickly, and there's a lot of old knowledge there that is, causing some challenges. So back to looking at our soil texture and soil organic matter. This chart is from the Cornell Health Assessment. So the solid line here is your coarse material. Those are your sands. The middle dashed long dash dot is the silts, and this dash line is the clay. So you can see the ranges of coarse would be organic matter of 2 to into the 4 range. So we're wanting, in this score of 60 and better, in the green zone here, it's 2 or better would be considered good. And as we increase, as we want to increase the organic matter, we have to consider soil texture. So as we move from a sandy to a silt to a clay, our target, our good zone for organic matter increases. So that's why it's very important to look at your soil texture before determining whether a score of 3 is in your target zone, but also referring back to your unmanaged area as your target, that your unmanaged area is your potential. So why is it that the organic matter in many of your unmanaged zones are like in the range of 5 to 10? And they're not even on this chart. And let's go to this one first. And it has to do with what management has, the impact of management in terms of depleting soil organic matter over time. So if we consider this just in terms of proportion of what makes up the soil organic matter percentage in the overall volume. So we have a big container of soil, and a small container within that is organic matter. And then within that small container, there's a difference in terms of that organic matter. Okay? Hopefully you're following. So within that organic matter, a large proportion of that is legacy organic matter that has built over a long period of time. And then the smaller portion is more recent additions. So some people would call it, there's the dead. So it's been dead organisms for a long period of time, and they've been entombed in the soil where the biology can no longer break them down. And the other part is still available for chewing up, breaking up, perhaps synthesizing if you have the biology there, and perhaps risking oxidation if there's disturbances and the management impact. So this is from a slide share from Joel Glover, who's got some excellent materials available. So older knowledge would have put us into in the organic matter in terms of what we're measuring. There's a 50% portion that is stabilized. So at the bottom of the pyramid, the big part of the pool. And then there's a 33 to 50%, that's the decomposing or active part, and then there's, the material that's more fresh. And research that the University of Minnesota has done has indicated that's not the case. After long-term agriculture, the large portion is the legacy, and the smaller portion is the material that can still be activated, can be still acted on by organisms. So if they get hungry, they could eat that carbon, or if you're losing soil, you could be losing that part. It means that as you're measuring more and more of the legacy. And so when you're measuring the unmanaged area and that number is high, you have a possibility that you're actually have more legacy long-term soil organic matter than you have in what is in the unmanaged area. So how do we know what the quality of that organic matter is? So are you following that? We're measuring something, but the proportion of what we're measuring is changing because the soil has degraded and we've actually lost this biologically active and recent, and we're starting to mine this part. We're starting to degrade this part. That's what we will have left, and the other side of that is that management changes can actually improve this. So when people say organic matter takes a long period of time, that's this component. This component, in terms of the biologically activated, can be added to. Management changes can change the number. I'm going to skip this part just because you can refer to it. It's just that organic matter has a lot of functions in the soil in terms of why it's important. I guess I'm not going to skip this part. It's important because it has so many multifunctions in terms of biology and sort of, it's a food source, it's an energy source, it's a home, it's a habitat, but it's contributing to the overall resiliency of the soil, and the resiliency is how much stress that it can take. And then there's a physical component in terms of keeping that soil together. And then the chemistry component is in terms of the cation exchange capacity and the ability to buffer changes in the pH and to be able to make complexes with the minerals. So balancing the inputs and the losses is, I suggest, is a good way of looking at it. And on balance, we want to increase our additions and know that different quality of those additions are important. And we want to minimize the losses. So the losses are, we're taking residues away, and we're also maybe leaving residues on the surface that are being oxidized, and there isn't sufficient biology or the type of biology to actually decompose those and incorporate them into the soil organic matter, so the active living component. And when we are adding the organic matter, the carbon material, we're providing living organisms food and energy, and they're able to create create compounds of nutrients, the soil aggregation, and then they're also able to transform that carbon into longer term carbon. So we want productivity to improve and our profits to improve, but at the same time, we're wanting our soil organic matter to improve. So two of the pieces of information that are on your data sheet are active carbon and soil respiration. And this is the interrelationship between the carbon process and the microbial process. So the soil active carbon is in units of ppm, parts per million. And so relative to the whole volume, it's the available food and energy for microbes. That's what they're going to have there to sustain them. That's your workforce. So the soil is mixed with potassium permanganate. And that causes a color change. And depending upon the degree of color change, because the active carbon is oxidized, they'll get a measurement of the active carbon. The soil active carbon is a better reflection of management that promotes soil organic matter accumulation and long-term soil carbon sequestration. So after you've gone through the initial steps where I said, comparing your managed to unmanaged and your management zones to your soil texture, look at your active carbon in terms of what proportion is in the active carbon to get an idea if you are contributing to long-term soil carbon sequestration. So we're looking for indicators that are sensitive to management change so that we can adapt our management. This is more informative than just organic matter percentage. Soil respiration, or the Solvita, is also on your report. That is the units of milligrams per carbon dioxide per gram of soil. So they take a measured volume of soil and they put it in a little, they dry it and they put it in a little container and they re-wet it and then measure the carbon dioxide that's coming off. And the carbon dioxide changes the color of this little palette. And it gives an indication not what soil biology is there, but their relative abundance and their activity. So they're in the soil, they went dormant when it dried, and then when you re-wet it, they got activated, they're hungry, and they start to decompose the carbon so that they're taking carbon into their system and transforming it, mineralizing it, and then they'll be making other nutrients available to store those nutrients and to change the soil structure. So the higher the CO2 given off, the more abundant and more active the soil microbes are. That's a leading indicator. That gives you a very responsive indication of your management. So looking at change in soil respiration is something that would change more dynamic and in a shorter period of time than organic matter or the active carbon. So when you compare those, you get a better idea of what your management impact is in terms of your soil, and perhaps why your worst or your best are performing differently. Questions? I hope you have questions because
There is a question in the chat.
good.
Yeah, I can ask it in person too. I was just
Thank you.
turning on
Hi,
all the
Randy.
switches. Hi there. So our standard testing in the West here, we're using Element Labs, and we haven't been doing our biological testing yet, and that's something I want to do. So when you're testing for active carbon and soil respiration, are you doing that through Cara or another lab?
That's through Kara.
Okay, good. So I've got to see if we have a Western equivalent, otherwise start using Cara. We do some samples in Manitoba through central testing, but certainly we want to step up our testing on our demonstration plot here. So it makes a lot of sense, and it's giving me a lot of food for thought. Thank you.
And Randy, for
Yep.
reference, Cara Labs is based out of Alberta. So it
Oh,
is,
it's
yeah.
closer. Yeah.
And they do partner with University of Alberta too for some of their stuff. So yeah, it's right in the West.
Okay, perfect. Thank you.
Yeah, and I'm correct in that, Dorothea. Those were done by Kara, right?
Yeah, that's correct.
Okay. I see that the Kara report, I mean, all of the biology was done at Kara, and their report, which you have, is quite extensive, and basically it breaks down into, microbial biomass in terms of bacterial or fungal, and then breaking out various microbes in terms of their type and their ratio and color and diameter. And um and I think Kara also made a presentation at Saskatoon. So when I look across, predominant, there's a higher biomass of bacterial organisms and relative to fungal organisms. And there is some correlation in terms of managed and unmanaged. And I think I I think there's specific ratios of bacterial to fungal that we're looking for. Um, and I'm still working at educating myself. So in terms of relying on Kara to do that interpretation and the reports, um, and comparing those, I think that's a next aspect here in terms of making use of those. They're very helpful in terms of, well, when you come to trying to change the overall active carbon and respiration. One of the ways of doing that is actually adding more biology. So, a number of folks here are using the teas, and I'm curious to see how much um, how much value we can see for that in terms of impact. And also, I really appreciated when when Allison said when she sees certain weeds growing or differences in crops, she'll actually, put the soil under the microscope and see what the ratios are. So yeah, this is where I say I need a lot better understanding myself. So I think, you know, when we see the biology and figure out what targets we're working for and get an idea of management changes, it's a lot more complex for me at this time. So, you know, start with an area that you're comfortable with and build on that. Um, and you know, put down at least something you understand is you know, constraining. Like if your compaction levels were higher than you would like to see them. What is your target? What is your goal? Because if we don't put something on paper and then, you know, go towards trying to implement that, we'll still be, you know, swirling around. Um, and and then, you know, step into that. And so, you know, maybe taking, when you're looking at what you're going to do for your new year, is at least taking one little area as your, you know, experiment or your trial area, and focus on that piece.